Hello students, in this video we are going to start about the second part of our biological classification, the chapter 2 class 11, and in the last class or the last part we already discussed about the classification of the living organisms, right? In that we studied about the earlier classification, then 2 kingdom classification, then 5 kingdom classification, 3 domain or 6 kingdom classifications. And in this video we are going to talk about the kingdom Monera. That's the first kingdom, that's the kingdom Monera. Okay, that is kingdom Monera. We already learned that it contains the prokaryotes, that is prokaryotes. That is a kingdom Monera is a made up of prokaryotes. Prokaryotes means the word itself, we can, pro means false or absence, karyo means nucleus. There is the absence of true nucleus. These prokaryotes are the group of organisms that is lack true nucleus. Pro means false or absent or lack or no. Karyo means carry on, that is carry on, that is carry on meaning nucleus. There is an absence of nucleus. So the kingdom Monera is a biological kingdom that is made up of prokaryotes, that is organism belonging to this group. Organism belonging to this kingdom do not contain true nucleus. That is, kingdom Monera includes the prokaryotes, most of them are the prokaryotes. That means they do not contain true nucleus. And their DNA, that means true nucleus means we don't have the proper nuclear membrane. There is no absence of nuclear envelope is absent. There is absence of nuclear membrane or nuclear envelope is absent. Their DNA is, that means their DNA is not enclosed within the nucleus. That is naked DNA. That is DNA is present, that is actually they are not enclosed within the nuclear membrane. As there is an absence of nuclear membrane, their DNA is not enclosed within the nucleus. So, it's a naked DNA. And this taxon Monera was first proposed as a phylum by the Ernst Haeckel in the year 1866. He was the first person, that's Ernst Haeckel was the first person who was proposed this taxon Monera as a phylum. So, in this part, that is a Monera is a biological kingdom that is made up of the prokaryote. Pro means the false, no or absent. Karyot means or carry on means nucleus. Absence of nucleus, that means these organisms belonging to this kingdom do not contain true nucleus. That means their DNA is not enclosed within the nucleus. That means they have the naked DNA. A nuclear envelope or nuclear membrane is absent in these organisms. And the taxon Monera was first proposed as a phylum by the Ernst Haeckel in the year 1866. And they are the oldest microorganisms known on the earth. These Monerans are the oldest microorganisms that we know on our earth. They are the first life on the earth. And it is composed of unicellular organisms, that is, they means a single celled organisms are, most of them are or all are single celled organisms, they are unicellular. And is commonly found in the moist environment and is also found in the hot springs, hot seasons, snow, deep oceans or as a parasites in other organisms. They may be found or they are mostly found in the various diverse environment conditions. That means they can be seen in the moist environment, hot spring season, they can be seen in the cold winter, that is a snowy areas, deep oceans or as a parasites in the other organs. Parasite means those that lives on other organisms and getting the food or obtaining everything from the host is called as a parasite and causes the infection. Sometimes mostly they cause the infection. They are called parasites. And no membrane-bound organelles in the cytoplasm, and they are with a single cellular circular chromosomes. They have the single circular chromosome and their cytoplasm is devoid of membrane bound organelles. Devoid means absence. There is a no membrane bound organelles in the cytoplasm with a single circular chromosomes. And the reproduction is mainly by the asexual mode of reproduction, that is by binary fission. Their reproduction mode of reproduction is the asexual reproduction, that is by binary fissions. And most bacteria are classified under Monera. The Monera coming under the Monerans are the bacteria. So we studied that they are the prokaryotes, they are the absence of nucleus, true nucleus are absent, then naked DNA, there is no nuclear membrane is absent, and first Monerans is proposed as means Monerans is classified or is changed to the phylum Monera by the Ernst Haeckel in the year 1866, and these are one of the oldest microorganisms that is known on the earth, and they are the single cellular organism or the unicellular, and they are found mostly in very diverse environmental conditions like moist environments, hot springs, snows, deep oceans, and sometimes as a parasite in other organisms. And there is a devoid of membrane bound organelles, there is a no membrane bound organelles in the cytoplasm, and there is a single circular chromosomes. And the reproduction is mainly by the asexual mode of reproduction, that is by binary fission, and the most bacteria are classified under kingdom Monera. And here the characteristic of Monerans, and the important characteristic of Monera are, Monerans are unicellular, that we already discussed in the last slide, that is a unicellular, that is single celled, and the DNA is naked, right, there is no true nuclear, that is not bounded by the nuclear membrane, as it is there is absence of nuclear membrane, there the naked DNA. That's a single circular chromosome is present in these organisms, and they contain 70s ribosomes in their cytoplasm. The cytoplasm is devoid of the membrane bound organelles, right? That is mitochondria, lysozymes, plastids, Golgi apparatus, then endoplasmic reticulum, centrosomes, etc. are absent. That is a cell membrane bound organelles are absent in the cytoplasm, but there is a 70s ribosomes and simple chromatophores are present in the cytoplasm of these Monerans. That is important characteristic of Monerans are unicellular, 70s ribosomes and a simple chromatophores are present. The DNA is naked and is not bounded by the nuclear membrane. Membrane bound organelles like mitochondria, lysozymes, plastids, Golgi apparatus, endoplasmic reticulum and the centrosomes are absent. And they reproduce by asexual mode of reproduction, asexually, that is by binary fissions or by budding. That is the mode of reproduction is the asexual mode of reproduction, or that is by binary fission or budding. And the cell wall is a rigid, their cell wall is rigid and is made up of peptidoglycan, right? Bacteria cell wall is mainly made up of the peptidoglycan, and they are the rigid cell wall, and that give shape as well as the protections to the organisms. And there is a locomotory organ, that is a hair-like appendages, that is a long elongated hair-like appendages that is starting from the base of the body, basal is attached to the organisms, and that helps for the movement of these organisms. There is a locomotory organ, that is for the movement. They are called the flagella, and these are the environmental decomposers. These Monerans are the environmental decomposers, and they show different mode of nutrition such as autotrophic, self preparing the food, parasitic, lives on the others and get the food, heterotrophic, also they are depending on the others for their food, then saprophytic, that feeds on the dead organic matters. So what are these are, that is they reproduce asexually by binary fissions or by budding. The cell wall, they have the rigid cell wall that is made up of the peptidoglycan, and the locomotory organ of these organisms is a flagella, and they are also called as environment decomposers. And various modes of nutrition, that is mode of nutrition they are attaining is that autotrophic, heterotrophic, parasitic as well as the saprophytic. So these are the characteristic of Monerans, that is they are unicellular, single celled, then they are the prokaryotic, that is a naked DNA is present, there is 70s ribosomes and a chromatophore in their cytoplasm, and the cytoplasm is devoid of the membrane bound organelles like Golgi apparatus, mitochondria, lysozymes, etc. and they the naked DNA is present, and there is a reproduce asexually by means of budding as well as the binary fission. Cell wall is rigid and that gives shape and protection to the organisms, and that is made up of peptidoglycan, and the locomotory organ of these groups are the flagellum, and they are also called as the environment decomposers. Various modes of nutrition are, they are parasitic, saprophytic, autotrophic, heterotrophic, etc. And next is the bacteria. They are the sole member of this kingdom. I already told you that most of the bacteria are grouped in the kingdom Monera, right? That's a sole member of the kingdom Monera is a bacteria. And they are most abundant microorganisms. There are numerous infinite microorganisms are present on our planet, right? So bacteria are the most abundant microorganisms and are present almost everywhere and can survive in the diverse different types of environment, diverse climatic condition, diverse environmental temperature conditions. That is almost everywhere their presence can be found and can survive in diverse environment conditions. And the numerous bacteria are present in the soil. Soil bacteria are also there. And the structure of bacteria actually very simple, that is they don't have the true nucleus and few cell organelles, but they have very complex characteristic. Okay, they have very complex characteristic and they can be beneficial as well as harmful. Some, that is not all are pathogenic, not all are beneficial. So there are some helpful, that's well as some harmful organisms are there. Helpful, actually we know that there is a lactobacillus is a very useful lactic acid bacillus, right? Lactobacillus for preparation of the curd and all we are using lactobacillus. Lactobacillus are very good for the gut movement and the digestion. Then some of them are the E. coli is present in our body, that is also very helpful for others. Then harmful, that's a pathogenic organisms are that's like Bacillus anthracis, Streptococcus. These all will cause the infections to our body. Right, so they are the sole members of this kingdom Monera are the bacteria. Abundant microorganisms are present in this are the bacteria and are present almost everywhere and kind of survive in the various environmental conditions, and they are also present in the soil. Then the structure is actually very simple with the lack of nuclear membrane and the cell membrane bound organelles, but they have very complex characteristic and they can be beneficial as well as the harmful effects. And many of them lives as a parasite. Most of them are living as a parasite. They live on the other organisms and cause the infections or some kind of disturbances in them. That's called parasite. They are getting the nutrition from them, right? And they live extreme habitats such as hot springs, deserts, snow and deep oceans. They have adverse, we already told that extreme, the diverse environmental conditions they live, that they live extreme habitats such as hot springs, deserts, snow and deep oceans. Bacteria are usually surrounded, this bacteria, all the bacteria are surrounded by two protective covering, that is the outer cell wall and also inner cell membrane. That is a protective coating or covering of the bacteria are the outer cell wall, that's a rigid cell wall is there, and the inner cell membrane is there. And the bacterial cell wall is made up of the peptidoglycan, that is a rigid cell wall is there, that is helps in the protection of the organisms. That is rigid cell wall is present and it is made up of lipopolysaccharide or peptidoglycan. And some bacteria or in some of the pathogen, mainly for the pathogenic bacteria, is covered by a slime layer outside, a sticky layer outside its a cell wall, that is called as a capsule. And the main function of this capsule is the giving protection to the organism from the host immune system, right? Okay, that is their cell wall is actually rigid and it is made up of either peptidoglycan or lipopolysaccharide. And in some of the pathogenic bacteria or some of the bacteria, there may be a slime layer or outer jelly like layer is covered onto the cell wall or covering the cell wall, that is called as a capsule. And this capsule, the main function of the capsule is that they helps in the protection of this pathogenic bacteria from the host immune system. Okay, and the few bacteria, that's like mycoplasma, they do not have the cell wall, they are absence of cell wall, there is a cell wall deficient bacteria is called as a mycoplasma. That is, many of them, these bacteria that lives as a parasite, they live in extreme habitats such as hot spring, desert, snow and deep ocean. And the bacteria usually surrounded by a protective covering called as outer covering, that's a cell wall, and inner cell membrane is that surrounds the cytoplasm. And the bacterial cell wall, that is made up of lipopolysaccharide or peptidoglycan, and it's a rigid cell wall, gives a shape and protections to the organisms. And in some cases or in some pathogenic bacteria, its cell wall is covered by a slime layer or the sticky layer outside the cell wall. That's called a capsule. That main function of this capsule is the giving the protection to the organism from the host immune system. And some of the cell wall bacteria are cell wall deficient. There is an absence of cell wall. They are called as a mycoplasma. And there is some other appendages also present in this bacteria. That is, first one is the cell wall. In some cases there is a capsule present that helps in the protection. The next is a pili or pili, anyway you can pronounce. Pili, it's a short, a small thin hair-like appendages that actually covers all surface of the body, all over the body, and they are numerous in numbers. It's called pili. That is, a pili is a short hair-like appendages on the surface of the bacterial cells, covers the bacterial cells. And there is another hair-like appendages, long hair-like appendages that is called helps in the locomotion of the movement of the organism. That is called a flagella. That is, pili is a short hair-like or small hair-like appendages that is covering the cell surface or the bacterial cell surface, that is called as a pili. And there is another hair-like appendages or the long slender appendages that is for the movement. They are called a flagella. And they show diverse metabolic activities. That is, even though they have the simple structures, simple cell size, that is devoid of the nuclear membrane, then cell organelles bound, membrane bound organelles are absent, still they show diverse metabolic activity. And they may be autotrophs or heterotrophs, as we discussed, or in many lower classes we know that what is meant by autotrophs and heterotrophs, right? And majority of these bacteria are heterotrophic. That means they depends on other organisms or external organic matters or on dead organic matter for their food. That is they may be the nutrition mode of nutrition is either autotrophic or heterotrophic. Heterotrophic means majority are heterotrophic. That means they depend on the other organisms or external organic matters or on dead organic matters for their food. So this locomotory organ is called as a flagella. That's a slender hair-like appendages that is extending from one end over the both the pole of the bacterial body. And pili is a short hair-like appendages that covers all over the body and is numerous in number. And they show diverse metabolic activity. They may be autotroph or the heterotrophic. And heterotrophic means that depends on the other organisms or other organic materials or dead organic matter for their food. And some are autotrophs, right, that they synthesize their own food from the inorganic substances. They are autotrophs, that means that they synthesize their own food from inorganic substances. And they can be saprophytic. What means the saprophytic? They lives on the dead organic matter. Parasitic means that is living on the another organisms and cause infections or diseases and gets all the nutrients. So that is a parasite. Symbionts means what? They lives in as a symbiotic man. That is means without disturbing them, that is they are not without causing any infection. There is a symbiosis, lives in a mutual understanding. That is symbiosis. That is, some of them are autotrophs. Autotrophs means that they synthesize their own food from the inorganic substrates, and they can be saprophytic, that is living on the dead organic matter. Parasitic, living on the other organism and causes infection. Symbionts means they are getting nutrients from the host, but they are not causing any damage to food. That is a symbiotes. Both are beneficial actually. And they may be photosynthetic autotrophs or chemosynthetic autotrophs. That is, either photosynthetic autotrophs or chemosynthetic autotrophs are there, that they derive carbon and hydrogen from the atmospheric carbon dioxide, and also that is from hydrogen, hydrogen sulfide and ammonia. That is, they may be saprophytic, parasitic or symbiotic. Then in the autotrophs, they may be photosynthetically autotroph or chemosynthetic autotroph, that they derive carbon, that is chemosynthetic autotroph, they derive carbon and hydrogen from the atmospheric carbon dioxide and hydrogen, hydrogen sulfide as well as the ammonia. And this is a structure of a bacterial cell that you can see, as I told you, there is a pink color, this is a cell wall is there, right? That's a rigid cell wall. Outer to the cell wall there is a slime layer or mucoid layer called as the capsule present. Inside the capsule there is a membrane that surrounds the cytoplasm, that is called the cell membrane, and here the blue purple color, that is a cytoplasm, and an oval shape, that is a plasmid is present, and the chromosome, that is a single circular chromosome is present, and the numerous small hair like appendages you can see, that is called as a pili or the fimbriae, and the long hair like appendages or the slender appendage that helps in the locomotion is called as the flagella, and the inclusion bodies are present, and 78 ribosomes also can be seen, and these are the bacterial cell, that is cell wall is the rigid cell wall made up of peptidoglycan or lipopolysaccharide, and the cell wall is surrounded by in some bacteria or the pathogenic bacteria there is a slime layer or a jelly like substances that is covering onto the cell wall. They are called as a capsule that helps in the protection, and to the inner to the cell wall there is a cell membrane that covers the cytoplasm, and inside the cell membrane there is a cytoplasm. The cytoplasm that contains a single circular chromosome is there, then plasmid is there, then inclusion bodies and ribosomes can be seen, and outer to the cell wall there is a hair like appendage numerous in number, they are called as the pili or the fimbriae, and single slender hair like long elongated hair like appendages helps mainly for the locomotion, they are called as the flagella. This is a structure of the bacterial cell. And next is the classification of the bacteria. Bacteria are classified mainly based on their shapes. Okay, the bacteria are classified mainly based on the shape, and they occur different shapes as well as they exist in the different forms. That is arrangements. They may show different arrangements of the cells. That is bacteria are classified based on their shape, and they possess various shape, and also they arrange in the different the cells are arranged in the various forms like a single cells, as sometimes in pairs, that is a diplo forms, that is a chain forms can be seen, or the cluster forms can be seen. That is at various arrangements of the bacteria. Okay, and the first one is a cocci, that is a shape cocci, that means cocci is a spherical or oval shaped bacteria are called as a cocci, and coccus is a singular word and cocci is a plural form of that. Okay, that is cocci is a plural form and coccus is a singular word, and they are the spherical or oval shaped bacteria, and they can be micrococcus, that is a single cells, and diplococcus, diplo, that means in pairs, cells are arranged in pairs. Then tetracoccus means tetra, the meaning tetra means 4, that is cells are arranged in a group of 4. And streptococcus, that streptococcus means there may be the arrangement of cells in the rows or chains can be seen. And the case of staphylococcus, the cells are arranged in the grape-like cluster. That is a cluster arrangement can be seen in the case of staphylococcus, that is a grape-like clusters. That is based on the bacterial shape, bacteria classified based on their shape, and there may be various shapes and their arrangement is also different, that is in pairs, in clusters, in singles or in chains. And the first one is a cocci, they are spherical or oval shaped bacteria, and they may be arranged as a single, that is a micrococcus, single arrangement, diplococcus in pairs, then streptococcus in the chains, tetracoccus, that is a group of 4 cells, then staphylococcus, they are the group of, that is a grape-like clusters. Next is the bacilli, they are the rod-shaped bacteria with or without flagella. Okay, the bacteria, that bacilli, that or bacillus, they are the rod-shaped bacteria or it's a form of a rod. Okay, there is a rod-shaped bacteria with or without flagella or elongated shape of bacteria. But other one is the cocci is a circular, right, or the spherical shape. This one is a rod or elongated shape. Example is the Bacillus anthracis. In the case of cocci, that is streptococci, pneumococci, these are some of the examples of the coccus. And in the bacillus, that is the Bacillus anthracis, that is the rod-shaped bacteria with or without the flagella. They may be motile, non-motile. Okay, with the flagella means they may be motile. Motile means moving. Without flagella means non-motile, that is they do not exist any movement. Motility is absent in the without a flagella. And Vibrio, they are the comma shaped or the kidney shaped small bacteria. They have a curved body structure that is called comma shaped, and with the flagella at one end. Flagella is present at the one end. Example, Vibrio cholera, and that is causative agent of the cholera. And Bacillus anthracis, the bacillus, there is a causative agent of anthrax. Spirillum, they are the spiral or coiled form as a corkscrew shaped. They are the spiral or coiled corkscrew shaped bacteria, and they are rigid forms due to the spiral structures and bear flagella at one or both ends. In the some of the spirillum, you can see that they have flagella at both the ends or at either at one end or at both ends, may be single or in clusters. Example, spirillum and spirochetes are some of the examples of the spirillum. And some can be seen as filamentous bacteria. The body consists of small filament like a fungal mycelia. Example, Bigiota and Theotrix. That is a filamentous bacteria, that is a body that consisting of a small filament like a fungal mycelia. Example, Bigiota and Theotrix. And the stacked, that is a bacterium body surrounded by a stack, that is Colobacterium, and budded, the body of a bacterium is swollen at places, that is Hyphomicrobium. Okay, these are the Hyphomicrobium, that is a budded or a body is swollen at one places. Stacked means elongated, it is a stack like structure, Colobacterium. Filament also, there is a small filament like a fungal mycelium, that is Bigiota and Theotrix are the examples. These are the different shape of bacteria, that is a coccus, that is a single arrangement. Diplococcus means two, diplo means two. Then diplo encapsulated pneumococci, you can see that a capsule is present. This is a capsulated cocci, they are in pairs. Next is a staphylococci, there is a grape-like clusters can be seen. Streptococci, they are the chain-like arrangements. Then Sarcina, tetrad, that is a tetrad means arrangement of 4, that is a tetrad. And Sarcina, you can see the 8, group of 8. And in the case of bacilli, there is a cocobacilli. Cocobacilli means is either a coccus form or the bacilli form. It is actually a mixed form of the merged form of the cocci as well as the bacilli. They were neither cocci nor bacilli. And the bacillus, this is a single cell, that is a rod-shaped bacteria. Diplobacilli, there is a 2 bacillus are there. And the palisade means arrangement of this bacteria in the corner joining. And next is a streptococci, there can be seen the chains. And another form is a vibrio, that is you can see that vibrio, there is a comma shaped bacteria, they have the single flagella or at the one end, and there is a elongated fusobacterium can be seen. Then you can see budded or appendaged bacteria, that is a hyphae is there, stacked bacilli is there. Then this is a spirillum, that is a corkscrew shaped bacteria, this is a filamentous bacteria, and this is a corkscrew shaped bacteria. And the corkscrew form of the Borrelia burgdorferi, and this is a spirochetes. And the club rod is there, Corynebacterium is there, then helical forms are there, Helicobacter pylori. Then see clusters, the Staphylococcus aureus, they can be grape-like clusters. Clostridium botulinum, you can see that there is an bulging at one end, and this is a Klebsiella pneumonia, that you can see the Diplococcus, and the Clostridium tetani. And here is also, this is also a Diplococcus, that is a Streptococcus pneumonia. And Bordetella pertussis, that is, you can see that there is a capsule is there encapsulated. Then Neisseria gonorrhea, that is a Diplococci. Then Neisseria, this is also Neisseria gonorrhea. Then this is a E. coli and Salmonella, that is a bacilli. Then Vibrio cholera, that is a comma shape. Then Streptococcus pyogenes, the chained cocci. And the Bacillus cereus, this is a, what is that? This is a chained bacilli. And Helicobacter pylori, see the helical structure is there. Then Treponema pallidum, you can see the spiral or the corkscrew shaped can be seen in the image. And next is the classification of the kingdom Monera. Kingdom Monera classified into 2 subkingdoms, that is Archaebacteria and Eubacteria. And here comes the classification chart. You can see the flow chart is the kingdom Monera. They are divided into domain Archaea and domain Eubacteria. And coming under the domain Archaebacteria, that is a methanogens, halophiles and thermoacidophils. In the name itself we can methanogens, that is a methane gas production. Halophiles, they can live in the saline water container. Then thermoacidophils, they can survive or thrive the heat and acidic conditions. And next is the domain Eubacteria. Domain Eubacteria is also divided into bacteria, cyanobacteria, actinomycetes, rickettsiae, spirochetes and mycoplasma. This that is archaea is methanogens, halophiles and thermoacidophiles. Domain Eubacteria, that is bacteria, cyanobacteria, actinomycetes, mycoplasma, rickettsiae, spirochetes. And this is the structure of the Archaebacteria, moreover similar to the bacteria only there, because they are the prokaryotes, right? So that is a capsule is present, and the pilus or the pili you can see, the flagellum is there, cell wall is there, plasma membrane is there, nucleoid is there, ribosome is there, cytoplasm is there. They are devoid of the other cell bound organelles. And next is the Archaebacteria. Archaebacteria is also known as the oldest living organism. They are known as the ancient living fossils. That is, they are the oldest group of organisms, oldest living organisms on the earth, and they are also known as, they are named ancient living fossils. Which is the ancient living fossils is the Archaebacteria, and they belong to the kingdom Monera. If they ask about which kingdom Archaebacteria, their kingdom Monera. And why they are classified in the bacteria? They are classified in the bacteria because they resembled moreover to the bacteria when observed under the microscope. They are resembled. There is a differences is there. Difference are also there, but they resemble almost similar. Resemblance are there with the bacteria when they observe under the microscope. That is why they are classified under the kingdom Monera or coming under the bacteria. That is Archaebacteria. They are the oldest living organism on the earth. They are all known as ancient living fossils. They are coming under the kingdom Monera. Why they are classified in the bacteria? Because they are similar. They resemble with the bacteria when observed under the microscope. And they are halophiles. As we told, Archaebacteria, they are the halophiles. Domain Archaea, they are halophiles, that is extreme salty environment. They live in an extremely salty environment. Thermoacidophiles, that means they can live in area with high temperature, and also methanogens, that means live in a low oxygen marshy areas. That is, they are halophiles. These Archaebacteria are halophiles. They are extremophiles. That is, they live in a extreme conditions. They are not normal conditions. They are not living. They require extreme conditions for their life. That is, halophiles, high salt areas. Thermoacidophiles, high temperature. Methanogens, low oxygen, marshy areas they are living. And the cell wall structure of Archaebacteria is differ from the bacteria. Even though they are similar or they are coming under the bacteria, they are different in their cell wall structure. That is, they lack the peptidoglycan. Bacterial cell wall is mainly made up of the peptidoglycan, but in the Archaebacteria, they lack peptidoglycan in the cell wall and branched lipids in the plasma membrane. And because of this, they are responsible for their survival in the extreme conditions. That is, cell wall structure of bacteria differ from the other bacteria. Because of this, they are responsible for their survival in the extreme conditions. And Archaebacteria, they are called as a methanogens because they are present in the these methanogens, they usually present in the gut of the animals like cows and buffaloes, and are responsible for the production of methane from the dung of these animals. They are usually present in the gut of the animals like cows and buffaloes, and that these bacteria are responsible for the methanogens are responsible for the production of the methane, that is a biogas, that from the cow dung or the buffalo dung. Okay, example is a methanobacterium. Methanogen, the methanobacterium, they are responsible for the production of the methane gas from the or methane from the dung of the cow, means waste or the byproducts of these animals, and they are present mainly in the gut of these cows and buffaloes. And Archaebacteria such as Thermus aquaticus is used, that is Archaebacteria such as Thermus aquaticus, that is a extreme temperature condition, that is a thermophiles. Thermoacidophiles, that is Thermus aquaticus, is used to obtain Taq polymerase enzyme. That is Archaebacteria Thermus aquaticus. Okay, Thermus aquaticus is used to obtain what? Taq polymerase. That Taq polymerase enzyme is obtained from the Thermus aquaticus, that is an Archaebacteria, and this the Taq polymerase, that is used in the recombinant DNA technology. That is, the recombinant DNA technology uses a Taq polymerase enzyme. These Taq polymerase enzyme are obtained from the Thermus aquaticus. That is the name of the bacteria. Okay, that is Thermus aquaticus is a group of Archaebacteria, that is thermoacidophilous group. Okay, and the mode of nutrition is a autotrophic. And the important characteristic of Archaebacteria are they are obligate or facultative anaerobes. Obligate means they are the strict anaerobes, that is only live under the absence of oxygen, that is a strict anaerobe. Anaerobe means what is? There are bacteria divided into two groups based on the oxygen requirement. Aerobe is there, then anaerobe is there, and facultative anaerobe is there, that is obligate anaerobe. That is aerobe means that lives under the presence of oxygen. Then strict aerobe is there, they only lives in the presence of oxygen. Then anaerobe is there, they do not survive in the oxygen, they require the anaerobic conditions for their growth. Okay. Then facultative anaerobe means they can live in the presence as well as the absence of oxygen. Okay. Obligate anaerobe means they are strict anaerobes. They are obligate, sometimes they may be strict anaerobes or facultative. They can live in the presence as well as the absence of oxygen. They can live in the absence of oxygen, so they can undergo methanogenesis. That is the process, the methane gas is called methanogenesis. That is why, because they can live in the absence of oxygen. And the mode of reproduction is asexual mode of reproduction, that is by the binary fission. And the cell wall that gives the shape and support or the strength to the cell wall helps in the protection from the harsh conditions. And they have a rigid cell wall that lacks the peptidoglycan, that we already told in the previous slide that they lack the peptidoglycan. That is the difference from the bacteria. Bacterial cell wall contain lipopolysaccharide or the peptidoglycan, but in the case of Archaebacteria they lack the cell, that is peptidoglycan in their cell wall. And the composed of branched lipids in their plasma membrane. They have the branched lipids in their plasma membrane, but peptidoglycan is absent. But in the cell wall, there is a pseudomurin is present. That is a pseudomurin that is present in the cell wall of Archaebacteria. What is the function of this pseudomurin? They protect this from the lysozyme. The protection, the function of this pseudomurin is a protection. From where it is getting protection? From the lysozyme. What is lysozyme? It is an enzyme that released from the host immune system. That is a these are pathogenic bacteria that when these organisms enter the body, our immune system or the animals, any living organism, their immune system will release some enzyme that is called lysozymes. That what is the function of this lysozyme? That lies the cell wall of the pathogenic bacteria. Okay, that is, once a pathogen enters the body, our immune system releases the lysozyme. That is an enzyme that helps to lyse the cell wall of the pathogenic bacteria. And the presence of pseudomurin in the cell wall of this Archaebacteria that protects it from the lysozyme. Okay, they give the protection from the lysozyme. That is, lysozyme is an enzyme that is released by the host immune system which lies the cell wall of the pathogenic bacteria, and they can withstand high atmospheric pressures also, and they do not possess membrane bound organelles such as nuclei, endoplasmic reticulum, mitochondria, lysozymes or the chloroplast. These all things we discussed in the previous slides, but when coming under the Archaebacteria, we have to, when there is any questions coming about characteristic of Archaebacteria, you have to write these points, right? That is why I have included in here below this also. Okay. And the other characteristic are that its a cytoplasm is a thick and contain all the compound required for the metabolism and the nutrition. Its a cytoplasm is thick. Even though there is a membrane bound organelles are absent in cytoplasm, it's a thick cytoplasm that contain all the compounds required for the metabolism and the nutrition. And they are known as extremophiles. Why they are called extremophiles? Because they can survive acidic and alkaline aquatic environment and also in the temperature above the boiling point. Why they are called? These are the Archaebacteria. They are called as the extremophiles. Why they are called extremophiles? Extremophiles means they survive in the acidic, alkaline, aquatic and as well as the high temperature, that is the above boiling point. These all environmental conditions that can survive, this bacteria can survive. That is why they are called as the extremophiles. And they also contain plasmids. Okay, in the picture Archaebacteria, when I showed the diagram, I showed that the plasmids are there, right? So they contain the plasmids. What is the use of or the function of this plasmid? That helps the antibiotic resistance. What is meant by antibiotic resistance? Antibiotic resistance means that resist the effect of the antibiotics, because one is that they lack the cell wall. So most of the antibiotics that inhibit the cell wall of the bacteria, and another is that they contain the plasmid, and the function of this plasmid is that with the help of this plasmid they resist antibiotic. That is, antibiotics will have no effects on these organisms. They cannot kill these organisms because the plasmid is there that act as an antibiotic resistant. Okay, as it have antibiotic resistant enzymes, that is that plasmid contain antibiotic resistant enzymes. Because of this they helps in the antibiotic resistance, that is means a protective, it's a shield for this Archaebacteria group of these organisms, or this Archaebacteria as a plasmid is acting as a protective shield for this organism, because there is an enzyme present in this that helps in the antibiotic resistance. So that is about the kingdom Archaebacteria, that is coming under the kingdom Monera. Up to this I am going to talk in this video, and the next video we will talk about the which one, Eubacteria. Okay, because it is a lengthy video, I don't want to club up everything and we feel bored. Instead of that, I have cut shorten this topic into 2 videos, and in the next video this part of next part of the kingdom Monera is coming. I think you guys can understand, my videos and can follow this. If you are really likes my video, please share to your friends, and thank you for today.